Setting up a timed assay is important when you are wanting to measure the rate of a reaction over a given time period. It is also useful so that you can run multiple reactions over the same period. This method of setting up reactions will be particularly useful during the enzyme kinetics laboratory sessions. Here you will be varying different experimental parameters such as substrate concentration, temperature and pH, and measuring the effect on the rate of reaction catalysed by the enzyme acid phosphatase. This reaction being studied is the conversion of the colourless substrate para-nitrophenol phosphate to the coloured product para-nitrophenol. These tubes contain the enzyme buffer together with increasing concentrations of the enzyme substrate, so that we can investigate how substrate concentration affects the rate of reaction. To start with, all tubes are placed in a water bath at 37 degrees centigrade for around 10 minutes to allow the solutions to equilibrate. A separate tube containing the enzyme solution is also incubated. Here we want to measure the rate of conversion of substrate into product over a time period of 15 minutes. At time 0, we start the timer on the addition of 1 milliliter of enzyme solution to the first tube. After 30 seconds, 1 milliliter of enzyme is added to the second tube. After a further 30 seconds, 1 milliliter of enzyme is added to the third tube. This process can be continued at 30-second intervals until the reaction has been started in all tubes. After 15 minutes, the first tube will have been incubated with the enzyme for the correct amount of time. And in this experiment, the reaction is stopped by the addition of 3 millilitres of sodium hydroxide. After a further 30 seconds, the second tube will also have been incubated for exactly 15 minutes. Once again, the reaction is stopped with the addition of 3 millilitres of sodium hydroxide. This is repeated until all reactions have been incubated for the correct amount of time. The tube should be removed and placed in a rack. The absorbance of the solutions can now be measured, and the amount of product produced by the enzyme is determined using a calibration graph.